Summary of Campbell Biology, 12th edition, Chapter 1, Evolution, the Themes of Biology, and Scientific Inquiry. Biology is the scientific study of life, and this chapter uses evolution as the central lens for understanding everything that follows. The chapter introduces the major themes of biology, shows how evolution ties them together, and explains how scientific inquiry builds biological knowledge. Life can be studied across a hierarchy of levels. Atoms combine to form molecules. Molecules assemble into structures within cells, and cells themselves are the basic units of life. In multicellular organisms, similar cells form tissues, tissues make up organs, and organs work together within organ systems. Individual organisms belong to populations, which interact with other populations in communities. Communities and their physical environments form ecosystems, and all ecosystems together make up the biosphere. Living things share several properties. They exhibit order in their structures, use and transform energy, regulate internal conditions, grow and develop according to inherited information, respond to environmental stimuli, reproduce, and show evolutionary adaptation as populations change over generations. These properties together define what biologists recognize as life. A key theme is that new properties emerge at higher levels of organization. When parts interact in more complex ways, the resulting system shows features that are not present in the individual parts alone. This idea of emergent properties appears repeatedly in biology, from molecules in a cell to interactions among organisms in an ecosystem. Cells are central to the study of life. Prokaryotic cells, found in bacteria and archaea, lack a nucleus and membrane-bound organelles. Eukaryotic cells, found in plants, animals, fungi, and protists, contain a nucleus and internal compartments. This division reflects deep evolutionary history and shapes how different groups of organisms live and function. Another theme is the flow of information. In all known organisms, DNA stores genetic information. The sequence of DNA provides instructions for building proteins and guiding cellular activities. When organisms reproduce, DNA is passed to offspring. Gene expression, the process by which information in DNA leads to functional products, underlies traits and behaviors. Energy and matter are also central themes. Energy flows through ecosystems, usually entering as sunlight and leaving as heat, while matter cycles among organisms and the environment. Photosynthetic organisms capture light energy and convert it into chemical energy that can be used by themselves and by consumers that eat them. Decomposers recycle nutrients, ensuring that materials remain available for future generations. Interactions are another recurring theme. Within an organism, different systems coordinate and regulate each other. In ecosystems, organisms interact with other species and with non-living components such as water, soil, and climate. These interactions shape population sizes, community structure, and the response of ecosystems to change. Evolution provides the unifying explanation for biological diversity and similarity. Populations change over time as genetic variations and environmental pressures interact. Natural selection favors individuals with inherited traits that increase their success in a given environment. Over many generations, this process can lead to adaptation and the formation of new species. The shared traits of organisms reflect common ancestry, while differences reflect evolutionary divergence. The chapter also explains how science investigates the natural world. Scientific inquiry involves making observations, forming hypotheses, testing them through experiments or additional observations, and analyzing the results. A hypothesis must be testable and falsifiable. Well-supported explanations that account for a wide range of observations are called scientific theories. They are broader in scope than hypotheses and are supported by extensive evidence. Together, evolution, the themes of biology, and the process of scientific inquiry create a framework for understanding life. The rest of the book builds on this foundation. Study questions. How do emergent properties relate to the hierarchy of biological organization? Answer. Emergent properties are new characteristics that appear when components interact at higher levels of organization, such as cells forming tissues or organisms interacting in ecosystems, and they cannot be predicted by examining the parts in isolation. Why is DNA considered central to the theme of information in biology? Answer. DNA stores the genetic instructions that guide development, structure, and function. Through gene expression and inheritance, DNA controls traits and transfers biological information from one generation to the next. How does evolution unify the many different topics studied in biology? Answer. Evolution explains why organisms share fundamental similarities due to common ancestry, and why they differ due to adaptation and divergence over time, providing a single framework that connects all areas of biological study. Unit 1. The Chemistry of Life. Chapter 2. The Chemical Context of Life. The 12th edition underscores that biological processes arise from chemical interactions. This chapter reviews the elements essential to life, the structure of atoms, and how chemical bonds form the molecules that make up organisms. Living things consist mainly of a small group of elements, with carbon, hydrogen, oxygen, and nitrogen making up most of their mass. Elements combine chemically to form compounds with properties different from those of their individual components. This concept is important for understanding how molecular structure influences biological function. Atoms contain protons and neutrons in a nucleus, surrounded by electrons arranged in energy levels. The number of protons identifies the element, while the number and arrangement of electrons determine how atoms interact with others. Atoms with incomplete outer electron shells tend to form chemical bonds to achieve stability. The chapter outlines several types of bonds. Covalent bonds form when atoms share electrons, producing strong and stable structures found throughout biological molecules. Ionic bonds form when electrons are transferred from one atom to another, creating oppositely charged ions that attract each other. The 12th edition places special emphasis on weak interactions, such as hydrogen bonds and van der Waals forces, because these subtle attractions play crucial roles in stabilizing the shapes of large biological molecules. The polarity of molecules influences how they behave in biological systems. Polar molecules have uneven electron sharing, creating partial charges that interact well with water. Non-polar molecules lack these charges and do not mix with water, a property vital for membrane structure and many cellular processes. Chemical reactions rearrange matter by breaking and forming bonds. Reactants become products, but the total amount of matter remains constant. In living organisms, reactions occur within controlled environments and often require catalysts such as enzymes. The text highlights that reactions proceed toward states of lower energy and greater stability. By understanding the behavior of atoms and molecules, biologists gain insight into how cells build their structures, perform chemical reactions, and maintain life. Study questions. Why do covalent bonds form the backbone of most biological molecules? Answer: they are strong, stable bonds that allow complex molecules to maintain their shape and function under the conditions found in living cells. How does electron arrangement determine chemical reactivity? Answer: atoms with incomplete outer electron shells are more likely to form bonds because they seek to gain, lose, or share electrons to reach a more stable state. Why does molecular polarity matter in biological systems? Answer: polarity affects how molecules interact with water and with each other, influencing processes such as membrane formation, solubility, and molecular recognition. Chapter 3: Water and Life. The 12th edition emphasizes water as the foundation of life's chemistry. This chapter explains how the structure of water molecules leads to special properties that support biological processes at every scale. Water is a polar molecule. Oxygen holds shared electrons more strongly than hydrogen, creating partial, negative, and positive regions. These regions allow water molecules to form hydrogen bonds with one another. Although each bond is weak, the combined network is strong enough to influence the behavior of water in significant ways. One key property is cohesion, the tendency of water molecules to stick together. Cohesion helps pull water upward through plant vessels, enabling long-distance transport without mechanical pumping. Adhesion, water's attraction to other materials, helps water cling to cell walls and move against gravity. Water moderates temperature changes due to its high specific heat. It takes considerable energy to disrupt hydrogen bonds, so water heats and cools slowly. This stabilizes both climate and the internal environments of organisms. Water's high heat of vaporization allows it to absorb large amounts of heat during evaporation, making processes such as sweating and leaf transpiration effective cooling mechanisms. When water freezes, hydrogen bonds lock molecules into an open structure, making ice less dense than liquid water. Because ice floats, it insulates the water below and protects aquatic life in cold seasons. This behavior is unusual among substances and is essential for life in many environments. Water is also an excellent solvent, able to dissolve ions and polar molecules by surrounding them with its charged regions. This allows cells to carry out metabolism in solution. Non-polar molecules tend to avoid water and group together, a behavior that helps form cell membranes and contributes to protein folding. Finally, water molecules can split into hydrogen and hydroxide ions. Substances that influence the concentrations of these ions change pH. The 12th edition highlights how buffers help organisms maintain stable pH levels, preventing harmful shifts that could disrupt cellular function. Together, these properties explain why water is indispensable and why life on Earth relies on it so completely. Study questions. How does water's polarity lead to hydrogen bonding? And why is this important? Answer: polarity creates slight charges that attract neighboring molecules, forming hydrogen bonds. These bonds give water cohesion, temperature stability, and solvent capabilities essential for life. Why does water help stabilize environmental and biological temperatures? Answer: its high specific heat allows it to absorb or release large amounts of heat with only small changes in temperature, protecting organisms and ecosystems from rapid fluctuations. How does the lower density of ice support life in aquatic environments? Answer: ice floats, creating an insulating layer that prevents bodies of water from freezing solid, allowing organisms to survive beneath the surface during cold periods. Chapter 4. Carbon and the molecular diversity of life. Carbon is central to biology because it has a unique ability to form a vast range of molecules. This chapter explains why carbon-based compounds are the foundation of life and how variations in carbon structure create the enormous diversity seen in biological molecules. Carbon has 4 valence electrons, allowing it to form 4 covalent bonds with other atoms. This versatility lets carbon build molecules that are small or large, simple or complex. Carbon can bond with hydrogen, oxygen, nitrogen, sulfur, and many other elements, giving rise to compounds with widely different properties. It can form single, double, or triple bonds, and it can link into chains, rings, and branching networks. These structural possibilities make carbon the backbone of many biological molecules. Molecules containing carbon are called organic molecules. Even though they share carbon as a common element, they can differ greatly in size, shape, and behavior. The arrangement of carbon atoms creates a molecule's skeleton, and even small changes in structure can produce major differences in function. For example, a chain of carbon atoms may behave differently than a ring made from the same number of carbons. Isomers are molecules with the same molecular formula, but different structures. Structural isomers differ in how their atoms are connected. Cis-trans isomers differ in spatial arrangement around a double bond, and enantiomers are mirror image forms that cannot be superimposed. Enantiomers are especially important in biology because organisms often recognize only one version. These differences highlight how structure influences function. Carbon skeletons gain additional variety through functional groups, specific groups of atoms that attach to the carbon backbone and change the molecule's properties. The 12th edition focuses on several functional groups commonly found in biological molecules. Hydroxyl groups make molecules more soluble in water. Carbonyl groups help form sugars. Carboxyl groups act as acids. Amino groups behave as bases. Sulfhydryl groups help stabilize protein structure through cross-linking. Phosphate groups store and transfer energy, and methyl groups affect gene expression and molecular recognition. These groups play essential roles in shaping how molecules behave inside cells. Life's large molecules are built from smaller carbon-based units. Although the detailed mechanisms of polymer formation are covered in the next chapter, this chapter establishes that carbon's bonding patterns make it possible for organisms to construct carbohydrates, lipids, proteins, and nucleic acids. Without carbon's flexibility, these complex molecules, and therefore life itself, would not exist. Overall, carbon's bonding capacity, structural variability, and collaboration with functional groups create almost limitless molecular diversity. This diversity underlies the complexity and adaptability of living organisms. Study questions. Why is carbon able to form such a wide variety of biological molecules? Answer. Carbon can make 4 covalent bonds, allowing it to form chains, rings, and branching structures with different elements, producing many possible molecular shapes and functions. What are isomers, and why are they important in biology? Answer. Isomers have the same molecular formula, but different structures or spatial arrangements. These differences can drastically change a molecule's behavior and how organisms interact with it. How do functional groups influence the properties of organic molecules? Answer. Functional groups add specific chemical behaviors such as acidity, basicity, reactivity, or solubility allowing molecules with similar carbon skeletons to perform very different biological roles. Chapter 5. The structure and function of large biological molecules. Living organisms are built from four major classes of large molecules: carbohydrates, lipids, proteins, and nucleic acids. These molecules are essential for structure, energy storage, information transmission, and nearly every function required for life. This chapter explains how these macromolecules are assembled, how their structures determine their roles, and why their diversity is central to biological complexity. Most large biological molecules are polymers, long chains made by linking smaller building blocks. The process that joins these monomers is called a dehydration reaction, in which two molecules bond while releasing a molecule of water. Polymers can later be broken down through hydrolysis, a reaction that adds water to split bonds. These reversible processes allow cells to assemble, remodel, and recycle their molecular components as needed. Carbohydrates include sugars and the polymers built from them. Simple sugars, or monosaccharides, generally contain carbon, hydrogen, and oxygen in a roughly 1:2:1 ratio. Glucose is a common example. These sugars can be linked to form disaccharides such as sucrose or long polysaccharides. Polysaccharides serve various roles. Starch stores energy in plants, while glycogen stores energy in animals. Cellulose forms strong structural fibers in plant cell walls, and chitin provides external support in arthropods and fungal cell walls. Differences in how glucose subunits are arranged give these polysaccharides distinct properties, illustrating how structure determines function. Lipids differ from other macromolecules because they are not polymers. Their unifying feature is that they do not mix well with water. Fats, one major type of lipid, consist of glycerol attached to fatty acids. Saturated fatty acids have no double bonds and tend to be solid at room temperature, while unsaturated fatty acids contain double bonds that create bends, making them more fluid. Phospholipids, which have two fatty acids and a phosphate group, form the basic structure of cell membranes by arranging themselves into bilayers. Steroids, another lipid category, include cholesterol and many hormones. Their four-ring structure allows them to play roles in signaling and membrane stability. Proteins are perhaps the most diverse macromolecules. They perform countless tasks, including catalyzing reactions, providing structural support, transporting substances, coordinating responses, and defending against pathogens. Proteins are polymers of amino acids, and there are 20 kinds commonly used by cells. The sequence of amino acids determines the protein's shape, which in turn determines its function. Protein structure is described in 4 levels. Primary structure is the linear sequence of amino acids. Secondary structure includes coils and folds stabilized by hydrogen bonds. Tertiary structure is the overall three-dimensional shape formed by interactions among side chains. Some proteins also have quaternary structure, created when multiple polypeptides assemble into a functional unit. Even slight changes in amino acid sequence can alter protein shape and impair function. Nucleic acids store and transmit genetic information. DNA holds the instructions for building proteins, while RNA helps carry out those instructions. These molecules are polymers of nucleotides made of a sugar, a phosphate group, and a nitrogenous base. DNA uses 4 bases, arranged in sequences that encode hereditary information. The 2 strands of DNA form a double helix, paired through specific base interactions. RNA usually consists of a single strand, and can take on various shapes that allow it to participate in protein synthesis and other cellular processes. Together, carbohydrates, lipids, proteins, and nucleic acids form the molecular foundation of all living systems. Their structures and the ways they interact make life's complexity possible. Study questions. How do dehydration reactions and hydrolysis relate to building and breaking down macromolecules? Answer: dehydration reactions join monomers by removing water to form polymers, while hydrolysis adds water to split bonds, allowing polymers to be broken back into monomers. Why do small structural differences in polysaccharides lead to major differences in function? Answer: because the way glucose units are linked changes the molecule's shape and chemical behavior, producing materials used for energy storage, rigidity, or protection. Why is protein structure so important for protein function? Answer: a protein's function depends on its three-dimensional shape, and that shape results from interactions among amino acids. Even minor changes can alter folding and disrupt activity. Unit 2. The Cell, Chapter 6. A tour of the cell. Cells are the fundamental units of life, and understanding their structure is essential for understanding how living organisms function. This chapter introduces the tools used to study cells and describes the major features of both prokaryotic and eukaryotic cells. The study of cells advanced significantly with improvements in microscopy. Light microscopes use visible light to produce images and are useful for observing living cells and many of their internal structures. Electron microscopes provide far greater resolution by using beams of electrons, allowing scientists to see detailed structures such as membranes and organelles. While electron microscopy offers clarity, it requires specimens to be non-living, so different techniques serve different purposes. Cells are small because their surface area must be large enough relative to their volume to support material exchange. As a cell increases in size, its volume grows faster than its surface area. Smaller cells maintain more efficient exchange with their surroundings, supporting the chemical processes required for life. All cells share certain basic features. They are surrounded by a plasma membrane, contain cytosol, hold genetic material, and use ribosomes to build proteins. Despite these similarities, there is a major distinction between prokaryotic and eukaryotic cells. Prokaryotic cells, found in bacteria and archaea, lack membrane-bound organelles and have DNA concentrated in a region called the nucleoid. Eukaryotic cells, found in plants, animals, fungi, and protists, contain a nucleus and many internal compartments that allow specialized functions. Within a eukaryotic cell, the nucleus houses DNA and controls gene expression. The nuclear envelope separates it from the cytoplasm, and nuclear pores regulate transport between the two. Chromatin, a complex of DNA and proteins, organizes genetic material within the nucleus. Organelles carry out specific functions. The endomembrane system includes the endoplasmic reticulum, Golgi apparatus, lysosomes, and various transport vesicles. The rough endoplasmic reticulum produces proteins destined for secretion or membranes, while the smooth endoplasmic reticulum performs functions such as lipid synthesis. The Golgi apparatus modifies, sorts, and ships products from the endoplasmic reticulum. Lysosomes contain digestive enzymes that break down macromolecules and recycle cellular components. Energy transformation occurs in mitochondria and chloroplasts. Mitochondria perform cellular respiration, converting chemical energy from food into ATP. Chloroplasts, found in plants and algae, capture light energy and convert it into chemical energy through photosynthesis. Both organelles contain their own DNA and ribosomes, supporting the idea that they originated from ancient symbiotic relationships. The cytoskeleton provides structural support and enables movement. It consists of microtubules, microfilaments, and intermediate filaments. These components help maintain cell shape, anchor organelles, and guide vesicle movement. In some cells, structures such as cilia and flagella use microtubules to create motion. Plant cells possess additional structures not found in animal cells. The cell wall provides rigidity and protection. Central vacuoles store water and contribute to cell growth through pressure. Chloroplasts enable photosynthesis. These differences reflect how plants meet their structural and metabolic needs. Together, the structures of a cell form an integrated system. Each part contributes to overall function, illustrating how compartmentalization supports complexity and efficiency in eukaryotic life. Study questions. Why are cells limited in size? Answer: cells must maintain a high surface area to volume ratio to efficiently exchange materials with their environment. Larger cells cannot transport substances quickly enough to meet metabolic needs. What distinguishes eukaryotic cells from prokaryotic cells? Answer: eukaryotic cells contain a nucleus and membrane-bound organelles, while prokaryotic cells lack these compartments and organize DNA in a nucleoid region. How do mitochondria and chloroplasts support the idea of an endosymbiotic origin? Answer: both organelles contain their own DNA, ribosomes, and double membranes, suggesting they evolved from ancient prokaryotes that began living inside early eukaryotic cells. Chapter 7: Membrane Structure and Function. Biological membranes define the boundaries of cells and their internal compartments. This chapter explains how membranes are built, how they regulate movement of substances, and how their dynamic nature supports life. Cell membranes are composed mainly of a phospholipid bilayer with embedded proteins. Phospholipids have hydrophilic heads and hydrophobic tails, causing them to arrange into a double layer with tails inside and heads facing outward. This structure is fluid rather than rigid. Phospholipids move laterally and proteins drift within the membrane. The fluidity depends on temperature and lipid composition. Unsaturated fatty acids keep membranes flexible, while cholesterol moderates fluidity by preventing extremes. Proteins embedded in the membrane perform essential tasks. Some act as transporters, letting specific molecules enter or exit the cell. Others function as receptors that detect signals. Additional proteins anchor the cytoskeleton, form cell junctions, or catalyze reactions. Carbohydrates attached to lipids or proteins serve as recognition markers that help cells identify each other. Selective permeability is a central property of membranes. Non-polar molecules pass easily, while polar molecules and ions require transport proteins. Diffusion moves substances from areas of higher to lower concentration. When water diffuses across a membrane, the process is called osmosis. Differences in solute concentration across membranes can cause cells to gain or lose water, affecting their survival. Transport can be passive or active. Passive transport does not require energy and includes simple diffusion, facilitated diffusion through protein channels, and osmosis. Active transport uses energy, usually from ATP, to move substances against their concentration gradients. One example is the sodium-potassium pump, which maintains essential ion gradients in animal cells. Bulk transport moves large materials through the membrane by vesicles. Endocytosis brings materials into the cell by forming vesicles from the membrane, while exocytosis releases materials by fusing vesicles with the membrane. These processes support nutrient uptake, waste removal, and signal release. Together, membrane structure and function enable cells to regulate their internal environments and interact with the outside world. Study questions. Why is membrane fluidity important? Answer. Fluidity allows membrane proteins and lipids to move, enabling processes such as cell signaling, transport, and membrane repair. What makes membranes selectively permeable? Answer. The phospholipid bilayer restricts many molecules, and proteins provide specific pathways, allowing the cell to control which substances cross. How does active transport differ from passive transport? Answer. Active transport requires energy to move substances against concentration gradients, while passive transport relies on natural diffusion and needs no energy input. Chapter 8. An Introduction to Metabolism. Metabolism includes all the chemical reactions that take place within an organism. This chapter explains how organisms obtain and use energy, how reactions are organized, and how enzymes regulate metabolic pathways. Metabolic pathways consist of steps in which one molecule is transformed into another. Each step is catalyzed by a specific enzyme. Some pathways break down molecules to release energy, while others build complex molecules using energy. Catabolic pathways, such as the breakdown of glucose, release energy by degrading molecules. Anabolic pathways, such as the synthesis of proteins, require energy to construct larger structures. Energy exists in many forms. Kinetic energy involves motion, while potential energy is stored due to position or structure. Chemical energy is potential energy available in molecules. The first law of thermodynamics states that energy cannot be created or destroyed, only transformed. The second law states that every energy transformation increases the disorder, or entropy, of the universe. Living systems maintain order by using energy, but they increase environmental entropy in the process. Free energy measures the portion of a system's energy available to do work. Reactions with a negative change in free energy occur spontaneously, while reactions with a positive change require an input of energy. Cells use ATP to power many non-spontaneous reactions. ATP stores energy in its phosphate bonds, releasing it when one phosphate group is removed. Enzymes speed up reactions by lowering the activation energy, the barrier that must be overcome for a reaction to proceed. Each enzyme works on specific substrates and forms an enzyme-substrate complex. After the reaction, products are released, and the enzyme is available to catalyze again. Temperature, pH, and chemicals can influence enzyme activity. Cells regulate metabolism by controlling enzyme activity and by using feedback inhibition, in which the end product of a pathway slows or stops its own production. Metabolism is highly coordinated, allowing organisms to respond to changes and maintain a stable internal environment. Study questions. What is the difference between catabolic and anabolic pathways? Answer. Catabolic pathways break down molecules and release energy, while anabolic pathways build complex molecules and require energy. Why do cells need ATP? Answer. ATP provides usable energy that powers non-spontaneous reactions, transport, and mechanical work. How do enzymes increase the rate of reactions? Answer: they lower activation energy, stabilizing the transition state and allowing reactions to proceed more quickly. Chapter 9. Cellular respiration and fermentation. Cells extract energy from food molecules to power their activities. This chapter explains how glucose is broken down, how ATP is generated, and how cells manage energy when oxygen is limited. Cellular respiration is a series of catabolic pathways that release energy by breaking down organic molecules. It involves three major stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. Glycolysis occurs in the cytosol and splits glucose into two molecules of pyruvate. This process yields a small amount of ATP and transfers electrons to NAD+, forming NADH. If oxygen is present, pyruvate enters the mitochondrion, where it is converted into a molecule that enters the citric acid cycle. The cycle completes the breakdown of glucose by releasing carbon dioxide and capturing high-energy electrons in the form of NADH and FADH2. Oxidative phosphorylation produces most of the ATP. The electron transport chain, located in the inner mitochondrial membrane, transfers electrons through a series of proteins. As electrons move down the chain, energy is used to pump protons across the membrane, creating a gradient. ATP synthase uses this gradient to generate ATP in a process known as chemiosmosis. When oxygen is scarce, cells can still produce ATP through fermentation. In lactic acid fermentation, pyruvate is converted to lactate. In alcohol fermentation, pyruvate is converted to ethanol and carbon dioxide. Fermentation regenerates NAD+, allowing glycolysis to continue, though it produces far less ATP than aerobic respiration. Cellular respiration illustrates how cells harvest energy efficiently and adapt to changing environmental conditions. Study questions. What are the three main stages of cellular respiration? Answer: glycolysis, the citric acid cycle, and oxidative phosphorylation. How does the electron transport chain generate ATP? Answer: it creates a proton gradient across the membrane, and ATP synthase uses this gradient to synthesize ATP. Why is fermentation important when oxygen is unavailable? Answer: it regenerates NAD+, so glycolysis can continue producing ATP, allowing cells to survive temporarily without oxygen. Chapter 10. Photosynthesis. Photosynthesis is the process by which plants, algae, and certain prokaryotes convert light energy into chemical energy stored in sugars. This chapter explains how light energy is captured, how electrons flow through photosynthetic pathways, and how carbon dioxide is transformed into organic molecules. Photosynthesis occurs primarily in chloroplasts, which contain pigments such as chlorophyll. These pigments absorb light energy that excites electrons. Photosynthesis consists of two main stages: the light reactions and the Calvin cycle. The light reactions take place in the thylakoid membranes. Here, chlorophyll absorbs light, transferring energy to electrons that travel through an electron transport chain. Water molecules are split to replace these electrons, releasing oxygen as a byproduct. The electron transport chain in chloroplasts resembles the one in mitochondria, but it uses light instead of food molecules as the initial energy source. As electrons move through the chain, protons are pumped across the membrane, generating a gradient. ATP synthase uses this gradient to produce ATP. Electrons eventually reach NADP+, forming NADPH. Thus, the light reactions generate ATP and NADPH, the energy carriers needed for the next stage. The Calvin cycle occurs in the stroma and does not require light directly. It uses ATP and NADPH to convert carbon dioxide into sugars through a process called carbon fixation. The cycle begins when the enzyme Ribisco attaches carbon dioxide to a 5-carbon molecule, eventually producing a 3-carbon sugar that can be used to build glucose and other organic molecules. The Calvin cycle regenerates its starting materials, allowing it to continue as long as ATP, NADPH, and CO2 are available. Environmental conditions affect photosynthesis. Plants must balance carbon gain with water loss through stomata. Some plants use alternative carbon fixation pathways, C4 and KAM photosynthesis, to reduce water loss or adapt to hot, dry environments. Photosynthesis supports life by producing the oxygen we breathe and forming the foundation of most food chains. Study questions. What are the main products of the light reactions? Answer: ATP and NADPH, which store energy and reducing power needed for the Calvin cycle. How does the Calvin cycle differ from the light reactions? Answer: the Calvin cycle uses ATP and NADPH to convert CO2 into sugars, while the light reactions capture light energy to produce ATP and NADPH. Why do some plants use C4 or KAM pathways? Answer: these pathways help minimize water loss and reduce photorespiration in hot or dry environments. Chapter 11. Cell communication. Cell communication is essential for coordinating functions, responding to the environment, and maintaining homeostasis. This chapter describes how cells detect signals, how messages are transmitted inside cells, and how responses are regulated. Cells receive signals through molecules such as hormones, neurotransmitters, or local regulators. Signaling typically involves 3 stages: reception, transduction, and response. Reception occurs when a signaling molecule binds to a receptor protein. Receptors may be embedded in the plasma membrane or located within the cell, depending on whether the signal can cross the membrane. Signal transduction usually involves multiple steps, forming a pathway in which proteins are activated or modified. Many transduction pathways rely on phosphorylation, the addition of phosphate groups by protein kinases, which can trigger cascades of activation. Secondary messengers, such as CAMP or calcium ions, amplify the signal and distribute it throughout the cell. The response varies depending on the signal and cell type. It may involve regulating a gene, activating an enzyme, rearranging the cytoskeleton, or initiating cell division. Because multiple steps are involved, cells can fine-tune the strength and duration of a response. Cells also regulate signaling to prevent overstimulation. Mechanisms such as dephosphorylation, signal degradation, and receptor removal ensure that signals end when they are no longer needed. Communication among cells enables multicellular organisms to coordinate growth, respond to stress, fight infections, and maintain stable internal conditions. Study questions. What are the 3 stages of cell signaling? Answer: reception, transduction, and response. Why are phosphorylation cascades important? Answer: they amplify signals and allow precise regulation of many steps in a pathway. How do cells ensure that signaling ends appropriately? Answer: by deactivating proteins, removing phosphate groups, degrading signaling molecules, or reducing receptor availability. Chapter 12. The cell cycle. The cell cycle is the sequence of events by which a cell grows and divides. This chapter explains how cells duplicate their genetic material, how division is regulated, and how loss of control can lead to cancer. Most of the cell cycle is spent in interphase, which includes G1, S, and G2 phases. During G1, the cell grows and performs its normal functions. In the S phase, DNA is replicated, producing identical sister chromatids for each chromosome. In G2, the cell prepares for division by synthesizing proteins and organelles needed for mitosis. Mitosis is the process of dividing the genetic material into two identical sets. It consists of prophase, prometaphase, metaphase, anaphase, and telophase. During mitosis, chromosomes condense. The nuclear envelope breaks down. Microtubules attach to chromosomes, and sister chromatids separate into two groups. Cytokinesis divides the cytoplasm, forming two daughter cells. In animal cells, a cleavage furrow forms. In plant cells, a cell plate develops. The cell cycle is controlled by checkpoints that ensure each phase proceeds correctly. Regulatory proteins called cyclins bind to cyclin-dependent kinases to trigger transitions. If conditions are unfavorable or DNA is damaged, the cycle can pause for repairs. When regulation breaks down, cells may divide uncontrollably, forming tumors and leading to cancer. The cell cycle allows organisms to grow, replace damaged cells, and reproduce asexually. Precise control ensures genetic stability from one generation of cells to the next. Study questions. What happens during the S phase of the cell cycle? Answer. DNA is replicated, producing sister chromatids for each chromosome. What is the role of cyclins and CDKs? Answer. They regulate cell cycle progression by triggering transitions between phases. Why can defects in cell cycle regulation lead to cancer? Answer. When checkpoints fail, cells may divide uncontrollably, accumulating mutations and forming tumors. Unit 3. Genetics. Chapter 13. Meiosis and sexual life cycles. Sexual reproduction produces offspring with genetic variation, and meiosis is the process that makes this possible. This chapter explains how chromosomes behave during meiosis, how gametes form, and how genetic diversity arises. Organisms have life cycles that alternate between fertilization and meiosis. In humans and most animals, fertilization combines two haploid gametes to form a diploid zygote. The zygote divides by mitosis to produce the multicellular organism. To maintain chromosome number across generations, meiosis reduces the diploid chromosome set to haploid gametes. Meiosis consists of two consecutive divisions, meiosis I and meiosis II. Meiosis Y separates homologous chromosomes, while meiosis II separates sister chromatids. Before meiosis begins, DNA is replicated, producing chromosomes made of two sister chromatids. In meiosis I, homologous chromosomes pair in a process called synapsis. While paired, they may exchange segments through crossing over, creating recombinant chromosomes that increase genetic variation. During metaphase Y, homologous pairs line up in the center of the cell. Their orientation is random, contributing to independent assortment. Homologs then separate and move to opposite poles. Meiosis II resembles mitosis. Chromosomes line up individually, and sister chromatids separate. By the end of meiosis, 4 haploid cells form, each genetically distinct. Genetic variation arises through crossing over, independent assortment of chromosomes, and random fertilization. These sources of variation contribute to evolution by providing raw material for natural selection. Study questions. Why must meiosis reduce chromosome number? Answer. To ensure that fertilization restores the diploid number without doubling chromosomes in each generation. How does crossing over increase genetic variation? Answer. It exchanges DNA segments between homologous chromosomes, producing recombinant chromatids with new allele combinations. What is independent assortment? Answer. The random orientation of homologous chromosome pairs during metaphase 1, which creates many possible gamete combinations. Chapter 14. Mendel and the Gene Idea. This chapter introduces the basic principles of inheritance discovered by Gregor Mendel. His work with pea plants revealed predictable patterns of trait transmission and established the foundation for genetics. Mendel studied traits that appeared in contrasting forms, such as purple or white flowers. By crossing plants and tracking traits across generations, he discovered that heritable factors, now known as genes, exist in pairs. One form or allele may mask the expression of another, leading to dominant and recessive relationships. In Mendel's monohybrid crosses, the F1 generation showed only the dominant trait. When F1 individuals were crossed, the F2 generation displayed a 3:1 ratio of dominant to recessive phenotypes. Mendel concluded that alleles segregate during gamete formation, and offspring receive one allele from each parent. Dihybrid crosses involving two traits revealed a 9.331 phenotype ratio in the F2 generation. Mendel interpreted this as evidence for the independent assortment of genes located on different chromosomes. Modern genetics has expanded Mendel's ideas. Some traits show incomplete dominance, codominance, or multiple alleles. Many traits are influenced by multiple genes or environmental factors. Despite these complexities, Mendel's principles still form the basis for predicting inheritance patterns. Study questions. What does Mendel's law of segregation state? Answer. Alleles separate during gamete formation, so each gamete receives only one allele for each gene. What phenotype ratio typically appears in a monohybrid F2 generation? Answer. A 3:1 ratio of dominant to recessive traits. Why do dihybrid crosses show a 9:331 ratio? Answer. Because alleles for different genes assort independently when they are on different chromosomes. Chapter 15. The chromosomal basis of inheritance. Mendel's principles became more powerful when linked to chromosomes. This chapter explains how genes occupy specific positions on chromosomes, how chromosome behavior explains inheritance patterns, and how linkage affects genetic outcomes. Chromosomes carry genes in specific locations called loci. Thomas Hunt Morgan's work with fruit flies provided evidence that genes are located on chromosomes. He discovered sex-linked inheritance when a mutation for white eyes appeared in male flies more frequently than females, showing that the gene was on the X chromosome. Sex chromosomes play a major role in inheritance patterns. In humans, females have two X chromosomes, while males have one X and one Y. Genes on the X chromosome often show distinct inheritance patterns because males express any allele they inherit, even if it is recessive. Genes on the same chromosome are linked and tend to be inherited together. However, crossing over can separate linked genes, creating recombinant offspring. The frequency of recombination between two genes reflects their distance on the chromosome, allowing scientists to create genetic maps. Chromosomal abnormalities can alter inheritance. Non-disjunction, the failure of chromosomes to separate during meiosis, can produce gametes with abnormal numbers of chromosomes. Conditions such as Down syndrome, Turner syndrome, and Klinefelter syndrome result from these events. These discoveries link Mendelian inheritance to physical structures, confirming chromosomes as the vehicles of genetic information. Study questions. Why do sex-linked traits appear more often in males? Answer. Males have only one X chromosome, so recessive alleles on that chromosome are expressed without being masked by a second allele. How does crossing over create recombinant offspring? Answer. It exchanges DNA between homologous chromosomes, producing new allele combinations on each chromatid. What causes non-disjunction? Answer. Failure of homologs or sister chromatids to separate during meiosis, leading to abnormal chromosome numbers in gametes. Chapter 16. The molecular basis of inheritance. The discovery that DNA is the genetic material transformed biology. This chapter explains how DNA encodes hereditary information, how it is structured, and how it replicates. Early experiments showed that DNA, not protein, carries genetic information. Studies using bacteria and viruses demonstrated that DNA is the molecule transmitted during infection and transformation, firmly establishing its role. DNA consists of two strands forming a double helix. Each strand is made of nucleotides with a sugar, a phosphate group, and a nitrogenous base. The bases, adenine, thymine, cytosine, and guanine, pair specifically, adenine with thymine and cytosine with guanine. These complementary base pairs allow each strand to serve as a template during replication. DNA replication is semi-conservative. Each new molecule contains one old strand and one newly synthesized strand. Replication begins at origins where DNA unwinds. Enzymes play coordinated roles. Helicase separates strands, single-strand proteins stabilize them, primase lays down RNA primers, and DNA polymerase adds nucleotides to growing strands. One strand, the leading strand, is synthesized continuously. The lagging strand is synthesized in fragments called Okazaki fragments, later joined by DNA ligase. Replication also includes proofreading and repair. DNA polymerases correct errors during synthesis, and repair systems fix mismatches or damaged bases. Maintaining DNA integrity is essential for stable inheritance and proper cellular function. Study questions. Why is DNA replication described as semi-conservative? Answer. Because each new DNA molecule contains one original template strand and one newly synthesized strand. What role does helicase play in replication? Answer. It unwinds the DNA double helix, separating the two strands so replication can occur. Why is the lagging strand synthesized in fragments? Answer. Because DNA polymerase can only build in one direction, requiring discontinuous synthesis on the strand oriented away from the replication fork. Chapter 17. Gene expression. From gene to protein. Gene expression is the process by which DNA directs the synthesis of proteins. This chapter describes how genetic information is transcribed into RNA and how RNA is translated into protein. Gene expression has two main stages: transcription and translation. During transcription, RNA polymerase binds to a gene's promoter and synthesizes a complementary RNA strand. This RNA transcript carries the coding information needed to build a protein. In eukaryotes, transcription produces pre-mRNA, which undergoes processing. A modified cap is added to the 5' end, a poly-A tail to the 3' end, and non-coding introns are removed through splicing. The remaining exons are joined to form the final mRNA, which exits the nucleus through pores. Translation occurs in the cytoplasm on ribosomes. The mRNA sequence is read 3 bases at a time in units called codons. Each codon specifies an amino acid. Transfer RNA molecules bring amino acids to the ribosome, where anticodons pair with mRNA codons. The ribosome links amino acids together to form a polypeptide chain, which folds into a functional protein. Gene expression is regulated at multiple levels, including transcription initiation, RNA processing, translation efficiency, and protein modification. These controls allow cells to respond to signals and adjust their functions. Study questions. What are the 2 main stages of gene expression? Answer: transcription, which makes RNA from DNA, and translation, which builds proteins from RNA. Why is RNA processing necessary in eukaryotes? Answer: it removes non-coding introns and adds protective modifications, producing a mature mRNA suitable for translation. How does tRNA contribute to translation? Answer: tRNA carries specific amino acids and matches them to codons on the mRNA through its anticodon. Chapter 18. Regulation of gene expression. Cells must control which genes they express and when. This chapter explains how prokaryotic and eukaryotic cells regulate gene activity, allowing specialization, adaptation, and efficient use of resources. In prokaryotes, groups of related genes can be organized into operons. An operon includes a promoter, operator, and genes with coordinated functions. The TRP operon is normally active but can be turned off when its end product accumulates. The LAC operon is usually off but is activated when lactose is present. These mechanisms help bacteria conserve energy by producing proteins only when needed. Eukaryotic gene regulation is more complex. Chromatin structure affects accessibility. Tightly packed chromatin is less accessible, while modifications such as acetylation can loosen DNA, promoting transcription. Transcription factors are essential for initiating gene expression and often require multiple proteins working together at enhancers and promoters. RNA processing adds another level of control. Alternative splicing can produce different proteins from the same gene. RNA stability also influences how long transcripts remain available for translation. Translation can be controlled by regulatory proteins or RNA molecules. After translation, proteins may require modifications or may be targeted for degradation, enabling cells to adjust protein levels quickly. Non-coding RNAs, including microRNAs, play important roles in gene silencing by binding to mRNA and preventing translation or triggering degradation. These regulatory mechanisms allow organisms to develop specialized cell types, respond to environmental changes, and maintain stable internal conditions. Study questions. What is an operon? Answer: a group of genes under coordinated control of a single promoter and operator, allowing prokaryotes to regulate multiple genes together. How does chromatin structure influence gene expression? Answer: loosely packed chromatin is more accessible for transcription. while tightly packed chromatin reduces gene activity. What role do microRNAs play in gene regulation? Answer: they bind to mRNA molecules, blocking translation or causing the mRNA to be degraded. Chapter 19. Viruses. Viruses occupy a unique position in biology because they can replicate only inside host cells. This chapter explores viral structure, replication cycles, genetic variation, and their effects on organisms. A virus consists of genetic material, either DNA or RNA, enclosed in a protein coat called a capsid. Some viruses also have an envelope derived from the host membrane. Viral genomes vary widely in size and organization, from a few genes to several hundred. Viruses infect cells by binding to specific receptors on the host surface. Once inside, they use the host's machinery to replicate. Bacteriophages. Viruses that infect bacteria follow two common cycles. In the lytic cycle, the virus immediately replicates, producing new phages that burst the host cell. In the lysogenic cycle, viral DNA integrates into the host genome and is copied as the host divides. Environmental triggers may cause the viral DNA to exit the chromosome and enter the lytic cycle. Animal viruses show similar patterns. Some integrate into the host genome. Others replicate in the cytoplasm. Retroviruses carry reverse transcriptase, which converts viral RNA into DNA that inserts into the host genome. HIV is a well-known retrovirus. Viral evolution occurs rapidly because of high mutation rates, especially in RNA viruses. This leads to emerging diseases, new viral strains, and host jumps. Vaccines and antiviral drugs help prevent or slow infections, but viruses continue to adapt. Although viruses challenge traditional definitions of life, they play major roles in ecosystems, evolution, and human health. Study questions. Why are viruses not considered fully living organisms? Answer: they cannot reproduce independently and must use a host cell's machinery to replicate. What is the difference between the lytic and lysogenic cycles? Answer: the lytic cycle destroys the host cell immediately, while the lysogenic cycle integrates viral DNA into the host genome for later activation. Why do RNA viruses evolve so rapidly? Answer: their replication enzymes lack proofreading, leading to high mutation rates and rapid genetic change. Chapter 20. DNA Tools and Biotechnology. Biotechnology includes techniques that use biological systems or molecules to modify organisms, study genetics, or produce useful products. This chapter covers DNA manipulation, cloning, sequencing, and applications in medicine and agriculture. Recombinant DNA technology allows scientists to combine DNA from different sources. Restriction enzymes cut DNA at specific sequences, producing fragments that can be inserted into plasmids. These recombinant plasmids enter bacteria, which replicate and produce the desired genes or proteins. Polymerase chain reaction, or PCR, amplifies a specific DNA segment rapidly. By cycling through heating and cooling, PCR separates DNA strands and uses a special polymerase to build copies, generating millions of fragments from a small initial sample. DNA sequencing reveals the order of nucleotides in a genome. Modern technologies allow rapid sequencing of entire genomes, enabling comparisons among species and identifying genes linked to traits or diseases. Gene expression can be analyzed using techniques such as RNA sequencing, which reveals which genes are active in a cell. Researchers also use CRISPR-Cas systems to edit genomes precisely by cutting DNA at targeted locations. This powerful tool has transformed genetic engineering. Biotechnology has many applications. Genetic engineering produces crops with improved traits, such as pest resistance or enhanced nutrition. Medical uses include producing insulin, diagnosing diseases, and developing gene therapies. Ethical challenges involve safety, environmental impacts, and genetic privacy. Biotechnology continues to expand, offering new possibilities for research and applied science. Study questions. What is recombinant DNA? Answer. DNA formed by combining genetic material from different sources into a single molecule. Why is PCR useful in biotechnology? Answer. It quickly amplifies specific DNA sequences, enabling research, diagnostics, and forensic analysis. What makes CRISPR-Cas systems powerful gene editing tools? Answer. They can cut DNA at precise locations, allowing targeted modifications to genomes. Chapter 21. Genomes and their evolution. Genome studies explore the full set of genetic material in an organism. This chapter explains how genomes are sequenced, how they vary, and what genome comparisons reveal about evolution. A genome includes all of an organism's DNA, including coding and non-coding regions. Advances in sequencing technology have made genome projects faster and more affordable. Bioinformatics, the use of computational tools to analyze large datasets, is essential for interpreting genome data. Genome annotation identifies genes and regulatory sequences. Comparing genomes among species helps identify conserved regions that likely have important functions. Many genes are shared across distant organisms, reflecting common ancestry. Eukaryotic genomes vary greatly in size. Much of their DNA does not code for proteins, but non-coding DNA includes regulatory sequences, repetitive elements, and mobile genetic elements such as transposons. These elements can move within the genome and contribute to evolution by creating new combinations of DNA. Gene duplication is a major source of evolutionary innovation. Duplicated genes may diverge over time, gaining new functions or specializing. Multigene families, such as those that encode hemoglobin subunits, arise from repeated duplication events. Genome evolution reflects changes in sequence, structure, and arrangement. By comparing genomes, scientists trace evolutionary relationships, identify gene functions, and understand how organisms adapt. Study questions. What is the role of bioinformatics in genomics? Answer. It uses computational tools to identify genes, compare genomes, and analyze large-scale genetic data. How do transposons contribute to genome evolution? Answer. They move to new locations in the genome, altering DNA sequences and potentially creating new gene combinations. Why is gene duplication important? Answer. It provides extra copies of genes that can evolve new or specialized functions, increasing biological complexity. Unit 4. Mechanisms of Evolution. Chapter 22. Descent with Modification, a Darwinian View of Life. This chapter introduces evolution as the fundamental explanation for biological diversity. Charles Darwin proposed that species change over time through natural selection, and this idea forms the foundation of modern biology. Darwin observed that species show variations, and many traits are inherited. He also noted that more individuals are born than can survive, leading to competition. From these observations, he proposed that individuals with traits better suited to their environments tend to survive and reproduce. Over generations, favorable traits become more common, a process known as natural selection. Fossils provide evidence of gradual change, showing transitions between ancient and modern forms. Comparative anatomy reveals similarities among organisms, such as homologous structures inherited from a common ancestor. Embryological patterns and molecular similarities further support evolutionary relationships. Darwin's concept of descent with modification explains both unity and diversity. Organisms share characteristics because they descend from common ancestors, yet they differ because they adapt to different environments. Natural selection acts on heritable variation, leading to adaptive change. Today, evolutionary theory is supported by genetics, biochemistry, and observations of natural populations. Evolution is not a linear process, but a branching one, producing diverse species from shared lineages. Study questions. What observation led Darwin to propose natural selection? Answer: that individuals vary in their traits and more are born than can survive, creating competition that favors advantageous variations. What are homologous structures? Answer: traits inherited from a common ancestor that may differ in form, but share underlying similarity. Why does evolution explain both similarity and diversity among organisms? Answer: similarity comes from shared ancestry, while diversity results from adaptations to different environments. Chapter 23. The Evolution of Populations. Populations evolve through changes in allele frequencies over generations. This chapter explains genetic variation, mechanisms of change, and how populations adapt. Genetic variation arises from mutations, gene duplication, and sexual reproduction. Mutations create new alleles, while sexual reproduction shuffles existing variation through recombination and independent assortment. The Hardy-Weinberg principle describes a non-evolving population where allele frequencies remain constant. Conditions for equilibrium include no mutations, random mating, no natural selection, large population size, and no gene flow. Real populations rarely meet all conditions, so allele frequencies often change. Three major mechanisms drive evolutionary change: natural selection, genetic drift, and gene flow. Natural selection increases the frequency of beneficial traits. Genetic drift involves random changes, especially in small populations. Events such as bottlenecks or founder effects can drastically alter genetic diversity. Gene flow occurs when individuals migrate between populations, transferring alleles and reducing differences. Natural selection acts on phenotypes and can favor traits in different ways. Directional selection shifts the population toward one extreme. Disruptive selection favors both extremes, and stabilizing selection maintains intermediate phenotypes. Adaptive evolution results when natural selection increases traits that improve survival or reproduction. Study questions. What produces most of the variation seen in sexually reproducing populations? Answer: the reshuffling of alleles through meiosis and fertilization. What is genetic drift? Answer: random changes in allele frequencies, especially common in small populations. How does gene flow affect populations? Answer: it transfers alleles between populations, reducing genetic differences. Chapter 24. The origin of species speciation explains how new species arise. This chapter explores reproductive barriers, patterns of divergence, and the processes that generate biodiversity. A species is often defined as a group of populations whose members can interbreed and produce fertile offspring. Reproductive barriers prevent gene flow between species. Prezygotic barriers block mating or fertilization, while postzygotic barriers reduce the viability or fertility of hybrid offspring. Allopatric speciation occurs when populations become geographically isolated. Over time, mutations, natural selection, and drift cause divergence. If populations become reproductively isolated, they may form new species. Sympatric speciation occurs without geographical separation. It may result from habitat shifts, sexual selection, or changes in chromosome number, such as polyploidy in plants. Speciation can be gradual or rapid. Fossil evidence shows bursts of change and longer periods of stasis. Hybrid zones provide insight into ongoing speciation. Hybrids may be less fit, leading to reinforcement of barriers, or hybridization may lead to fusion or stability. Macroevolution encompasses broad patterns of evolution above the species level, including the emergence of new groups and the impact of major changes in the environment. Study questions. What distinguishes prezygotic from postzygotic barriers? Answer. Prezygotic barriers prevent mating or fertilization. Postzygotic barriers reduce hybrid survival or fertility. How does allopatric speciation occur? Answer. Populations become geographically separated and diverge genetically until reproductive isolation arises. Why are hybrid zones useful for studying speciation? Answer. They show interactions between diverging populations and reveal whether reproductive barriers are strengthening, weakening, or stable. Chapter 25. The history of life on Earth. Earth's history provides essential context for understanding biological diversity. This chapter describes how life originated, how major groups evolved, and how geological processes have shaped life. Life likely began through a series of chemical steps. Early Earth had conditions suitable for forming organic molecules. Laboratory models suggest that simple molecules could arise from inorganic components under energy-rich conditions. These molecules may have assembled into protocells, membrane-bound structures capable of maintaining internal environments. RNA is a likely candidate for the first genetic material because it can store information and catalyze reactions. The fossil record describes major transitions in life's history. Prokaryotes were Earth's first organisms and dominated for billions of years. Photosynthesis evolved and began producing oxygen, which accumulated in the atmosphere and altered Earth's chemistry. Eukaryotes arose later, formed through endosymbiotic events in which prokaryotes became permanent residents within host cells. Multicellular organisms eventually emerged and displayed increasing complexity. Life's history is punctuated by dramatic events. Plate tectonics rearranges continents, altering environments and influencing evolution. Mass extinctions have repeatedly reshaped biodiversity by eliminating large groups while opening opportunities for survivors. Adaptive radiations, rapid diversification events, often follow these extinctions or the evolution of key innovations. This chapter shows that Earth's history and life's history are deeply intertwined, with geological processes and biological evolution influencing each other. Study questions. Why is RNA considered a strong candidate for the first genetic material? Answer: because it can store information and act as a catalyst, allowing early molecules to replicate and evolve. What major event changed Earth's atmosphere and enabled aerobic life? Answer: the evolution of photosynthesis, which released oxygen into the atmosphere. How do mass extinctions influence evolution? Answer: they eliminate many species, opening ecological opportunities that allow surviving groups to diversify. Unit 5. The Evolutionary History of Biological Diversity. Chapter 26. Phylogeny and the Tree of Life. Phylogeny reconstructs evolutionary relationships among species. This chapter explains how scientists build evolutionary trees, interpret shared traits, and classify life. Systematics uses data from morphology, genetics, and biochemistry to infer relationships. A phylogenetic tree represents hypotheses about evolutionary history, showing how species diverged from common ancestors. Branch points represent divergence events, while sister taxa share a recent ancestor. Homologous structures arise through common ancestry and are key to reconstructing phylogeny. Analogous traits, by contrast, result from convergent evolution and do not indicate shared ancestry. Distinguishing between the two is essential for accurate trees. Cladistics groups organisms by shared, derived characters, traits unique to certain lineages. A clade includes an ancestor and all its descendants. Trees aim for parsimony, the simplest explanation consistent with the data. Modern phylogenies increasingly rely on DNA sequences. Molecular clocks estimate divergence times based on genetic change. These tools have reshaped classification, revealing that life is divided into 3 domains: bacteria, archaea, and eukarya. Classification is dynamic because new data can revise our understanding of relationships. Phylogeny provides a framework for studying evolutionary patterns and the unity and diversity of life. Study questions. What is a clade? Answer: a group consisting of an ancestor and all its descendants. How do homologous traits differ from analogous traits? Answer: homologous traits arise from shared ancestry. Analogous traits arise from independent adaptation to similar environments. Why has DNA become central to phylogeny? Answer: because genetic sequences provide precise information about evolutionary relationships and divergence times. Chapter 27. Bacteria and Archaea. Prokaryotes are the most abundant and diverse organisms on Earth. This chapter explores their structure, metabolism, reproduction, and ecological roles. Bacteria and Archaea share basic cell structures: a plasma membrane, cytosol, DNA in a nucleoid region, and ribosomes. Most have cell walls. Bacterial walls contain peptidoglycan, while archaeal walls vary in structure, contributing to differences in their biochemistry. Prokaryotes reproduce by binary fission, producing genetically identical cells. However, they generate variation through rapid mutation and genetic recombination processes such as transformation, transduction, and conjugation. These processes allow gene exchange even without sexual reproduction. Prokaryotes display remarkable metabolic diversity. Some are phototrophs, obtaining energy from light. Others are chemotrophs, using chemical sources. They may use organic or inorganic compounds as carbon sources. This diversity allows them to inhabit extreme environments, including hot springs, salt lakes, and deep-sea vents. Archaea include groups adapted to extreme conditions, such as thermophiles, halophiles, and methanogens. Bacteria include many familiar forms, including pathogens, but also beneficial species. Prokaryotes perform vital ecological functions. They decompose organic matter, recycle nutrients, fix nitrogen, and support ecosystems. Some form symbiotic relationships with plants and animals. They are also used in biotechnology for producing chemicals, treating wastewater, and engineering new biological tools. Study questions. Why do prokaryotes evolve quickly? Answer: they reproduce rapidly and accumulate mutations quickly, and they exchange genes through recombination processes. What distinguishes archaeal cell walls from bacterial cell walls? Answer: bacteria have peptidoglycan, while archaea lack peptidoglycan and use different structural materials. Why are prokaryotes essential for ecosystems? Answer: they recycle nutrients, decompose organic matter, fix nitrogen, and support food webs through key biochemical processes. Chapter 28. Protists. Protists are a diverse collection of mostly unicellular eukaryotes that do not fit neatly into the plant, animal, or fungal kingdoms. This chapter explains their diversity, evolutionary origins, and ecological roles. Protists share a eukaryotic cell structure with membrane-bound organelles, but they vary widely in nutrition, reproduction, and lifestyle. Some are photoautotrophs, others are heterotrophs, and many are mixotrophs that combine both strategies. Their diversity reflects multiple evolutionary lineages rather than a single ancestral group. Endosymbiosis played a key role in protist evolution. The origin of mitochondria and chloroplasts occurred through symbiotic relationships in which one organism lived inside another. Secondary endosymbiosis further expanded diversity when a eukaryotic cell engulfed another eukaryotic alga, producing new photosynthetic groups. Protists exist in several major clades. Excavates include species with modified mitochondria or unique flagella. Cellular, a large and diverse supergroup, includes diatoms, brown algae, dinoflagellates, and various parasites. Archaeoplastitida contains red algae, green algae, and land plant ancestors. Uniconta includes amoebas and the lineages leading to fungi and animals. Protists play essential ecological roles. Photosynthetic protists produce a significant portion of Earth's oxygen and support aquatic food webs. Heterotrophic protists recycle nutrients by consuming bacteria and other microorganisms. Some protists cause diseases such as malaria and dysentery. Despite their diversity, protists share the characteristic of being eukaryotes with versatile cellular structures adapted to many environments. Study questions. How did endosymbiosis contribute to protist diversity? Answer: it led to the origin of mitochondria and chloroplasts and later produced new protist lineages through secondary endosymbiosis. Why are protists not classified as a single kingdom? Answer: because they include multiple evolutionary lineages and do not form a single monophyletic group. What ecological roles do photosynthetic protists play? Answer: they produce oxygen and form the base of many aquatic food webs. Chapter 29. Plant diversity. How plants colonized land. The transition from water to land was one of the most significant events in the history of life. This chapter explains the adaptations that enabled plants to survive on land and describes the earliest plant groups. Land plants evolved from green algae. Fossil and molecular evidence points to charophyte algae as the closest relatives. Living on land offered advantages, such as more sunlight, more CO2, and fewer herbivores initially. However, it also presented challenges, including the risk of drying out. Plants developed several adaptations for terrestrial life. A waxy cuticle reduced water loss. Stomata allowed gas exchange while helping regulate moisture. Multicellular structures protected gametes and embryos. Vascular tissues, which would appear later, improved structural support and water transport. The earliest land plants were non-vascular and relied on moist environments. Bryophytes, mosses, liverworts, and hornworts lack extensive vascular tissue. They have dominant game-to-fight stages and require water for fertilization. Their small size reflects limited internal transport abilities. The appearance of vascular tissue marked an evolutionary shift. Early vascular plants had xylem and phloem for water and nutrient transport, allowing them to grow taller and colonize more habitats. The movement of plants onto land transformed Earth's ecosystems by stabilizing soils, altering atmospheric composition, and creating habitats for other organisms. Study questions. What evidence links land plants to green algae? Answer. They share key traits such as photosynthetic pigments, cell wall structure, and molecular similarities. Why was a cuticle important for early land plants? Answer. It reduced water loss in the dry terrestrial environment. Why are bryophytes restricted to moist areas? Answer. They lack vascular tissues and depend on water for sperm to reach eggs. Chapter 30, Plant Diversity 2, the Evolution of Seed Plants. Seed plants dominate most terrestrial ecosystems today. This chapter describes the evolution of seeds, pollen, and other adaptations that allowed plants to thrive on land. Seeds provide protection, nourishment, and improved dispersal for developing embryos. A seed contains an embryo, a food supply, and a protective coat. Seed formation required innovations in reproduction, including reduced gametophytes, pollen, and ovules. In seed plants, the gametophyte is microscopic and depends on the sporophyte for nutrients. Pollen grains contain male gametophytes and can travel without water, enabling fertilization across long distances. Ovules house the female gametophyte and develop into seeds after fertilization. Gymnosperms were the first seed plants. They include conifers, cycads, ginkgo, and netophytes. Gymnosperms dominated during the age of dinosaurs and are well adapted to dry conditions. Their seeds are exposed on cones rather than enclosed in ovaries. Angiosperms, the flowering plants, later became the most diverse plant group. Flowers attract pollinators, improving reproductive efficiency. Fruits develop from ovaries and aid in seed dispersal by animals, wind, or water. These interactions between plants and animals accelerated angiosperm diversification. Seed plants transform terrestrial environments, providing food, habitats, and stability for entire ecosystems. Study questions. What advantages do seeds provide? Answer. They protect embryos, supply nutrients, and allow dispersal across varied environments. How did pollen contribute to seed plant success? Answer. It allowed fertilization without water, enabling reproduction over long distances. What key innovations helped angiosperms diversify? Answer. Flowers that attract pollinators and fruits that enhance seed dispersal. Chapter 31. Fungi. Fungi are essential organisms in nearly every ecosystem, functioning as decomposers, symbiotic partners, and sometimes pathogens. This chapter explains fungal structure, reproduction, and ecological importance. Fungi are eukaryotes that absorb nutrients from their surroundings. Instead of ingesting food, they secrete enzymes that break down complex molecules into smaller compounds that can be absorbed. This external digestion allows them to decompose materials such as fallen leaves, dead animals, and wood. Most fungi grow as networks of filaments called hyphae. These filaments form a mycelium, which infiltrates the surface of the material the fungus feeds on. Hyphae may be divided into cells by septa or remain continuous. Their structure increases surface area, enhancing nutrient absorption. Fungi reproduce through spores, which can be produced sexually or asexually. Sexual reproduction typically involves the fusion of cytoplasm from two parent mycelia, followed later by the fusion of nuclei. This results in genetic recombination and the formation of spores that disperse to new environments. Asexual reproduction often involves the formation of spores through mitosis, allowing rapid colonization. Fungi form important symbiotic relationships. Mycorrhizal fungi associate with plant roots, improving water and nutrient uptake. Lichens consist of fungi living with algae or cyanobacteria, producing communities that colonize harsh environments. Some fungi are pathogens of plants or animals, while others contribute to human uses such as food production and medicine. Fungi play indispensable ecological roles by recycling nutrients and forming symbioses that support entire ecosystems. Study questions. What structural feature of fungi enhances their ability to absorb nutrients? Answer: hyphae form a large mycelial network that provides extensive surface area for absorption. How do fungi obtain nutrients? Answer: they secrete digestive enzymes externally and absorb the resulting smaller molecules. What is a mycorrhizal association? Answer: a symbiosis in which fungi help plant roots absorb water and nutrients in exchange for carbohydrates. Chapter 32. An overview of animal diversity. Animals are a diverse group of multicellular eukaryotes with specialized tissues and complex developmental patterns. This chapter introduces their origins, key characteristics, and main branches of the animal tree of life. Animals are heterotrophs that ingest food. Their bodies are composed of tissues derived from embryonic layers. Most animals have muscles and nervous tissue, enabling movement and rapid response. Reproduction is primarily sexual, with a dominant diploid stage. The fossil record shows that animals originated more than 600 million years ago. The Cambrian explosion marked a major diversification event, producing many of the body plans seen today. Several factors contributed to this rapid evolution, including increased oxygen, predator-prey interactions, and changes in developmental genes. Animals are classified based on body symmetry, tissue organization, and early developmental patterns. Sponges lack true tissues, while other animals have distinct tissue layers. Bilateral symmetry, seen in most animals, allows for cephalization and directional movement. Diploblastic animals have 2 tissue layers, while triploblastic animals have 3. The development of a body cavity, or coelom, provides space for organs and improves movement. Animals also differ in their embryonic development. Protostomes typically exhibit spiral cleavage and form the mouth first, while deuterostomes have radial cleavage and form the anus first. These patterns allow biologists to group animals into major clades, including Lophotrochozoa, Ecdysozoa, and Deuterostomia, each containing diverse phyla. Study questions. What key characteristics define animals? Answer: they are multicellular, ingestive heterotrophs with specialized tissues and complex development. Why was the Cambrian explosion significant? Answer: it produced a rapid diversification of animal body plans, establishing many major lineages. What is the difference between protostomes and deuterostomes? Answer: protostomes form the mouth first during development, while deuterostomes form the anus first. Chapter 33. An Introduction to Invertebrates. Invertebrates make up the vast majority of animal diversity. This chapter explores major invertebrate groups, their characteristics, and their evolutionary relationships. Sponges are among the simplest animals. They lack true tissues and feed by filtering water through pores. Cnidarians, including jellyfish and corals, have radial symmetry and specialized stinging cells. They have two main body forms, polyp and medusa. Lophotrochozoa includes flatworms, mollusks, and annelids. Flatworms have simple bodies without a coelom. Mollusks include snails, clams, and octopuses, and share a body plan with a foot, visceral mass, and mantle. Annelids are segmented worms with repeated body units. Ecdysozoa includes animals that shed an external covering in a process called molting. Nematodes are roundworms found in nearly all habitats. Arthropods, the most diverse animal phylum, include insects, crustaceans, and arachnids. They have exoskeletons, segmented bodies, and jointed appendages, enabling success in land, water, and air. Echinoderms, although invertebrates, are closely related to vertebrates. They exhibit radial symmetry as adults, but have bilateral larvae. Sea stars, urchins, and their relatives have water-vascular systems for movement and feeding. Invertebrates demonstrate a remarkable range of forms and adaptations, highlighting the evolutionary creativity of animal life. Study questions. What body feature allows arthropods to be highly successful? Answer: their exoskeleton and jointed appendages enable protection, movement, and specialization. What defines cnidarians? Answer: they have radial symmetry, stinging cells, and two main body forms, polyp and medusa. Why are echinoderms considered close relatives of vertebrates? Answer: they share developmental patterns characteristic of deuterostomes. Chapter 34. The Origin and Evolution of Vertebrates. Vertebrates are a diverse group within the chordates, defined by a backbone and complex nervous systems. This chapter describes their early evolution and the major groups that arose from their lineage. Chordates share four key features at some point in development: a notochord, a hollow dorsal nerve cord, pharyngeal slits, and a muscular post-anal tail. Vertebrates differ from other chordates by having vertebrae that enclose the nerve cord and a skull that protects the brain. The earliest vertebrates were jawless fish that lived more than 500 million years ago. The evolution of jaws was a major innovation, allowing more efficient feeding. Cartilaginous fish, such as sharks, belong to one major lineage of jawed vertebrates. Ray-finned fishes and lobe-finned fishes form two large groups of bony vertebrates. Lobe-finned fishes gave rise to tetrapods, vertebrates with four limbs. The transition from water to land involved adaptations such as stronger limbs, lungs, and supportive skeletons. Amphibians were the first tetrapods to spend part of their lives on land. They still depend on moist environments for reproduction. Reptiles and mammals evolved from amniotes, which produce amniotic eggs that protect embryos on land. Reptiles include birds, which are feathered descendants of theropod dinosaurs. Mammals evolved distinctive features such as hair, mammary glands, and specialized teeth. They diversified into monotremes, marsupials, and placental mammals. Vertebrate evolution shows repeated innovation and diversification as organisms adapted to new environments and challenges. Study questions. What key features distinguish chordates? Answer: a notochord, a dorsal nerve cord, pharyngeal slits, and a post-anal tail. Why was the evolution of jaws important? Answer: it allowed vertebrates to capture and process food more effectively, expanding ecological opportunities. What adaptation enabled amniotes to succeed on land? Answer: the amniotic egg, which protects embryos from dehydration and allows development away from water. Unit 6. Plant Form and Function. Chapter 35. Vascular Plant Structure, Growth and Development. Plants display remarkable structural diversity, but they share core organizational principles. This chapter explains how vascular plants are built, how they grow, and how developmental processes shape their bodies. Vascular plants have three main organs: roots, stems, and leaves. Roots anchor the plant and absorb water and minerals. Stems support leaves and transport substances through vascular tissues. Leaves are the main sites of photosynthesis. Tissues form the building blocks of organs. Dermal tissue provides protection. Vascular tissue, xylem and phloem, transports water, minerals, and sugars. Ground tissue carries out functions such as photosynthesis, storage, and support. Plant growth arises from meristems, regions of unspecialized cells that divide throughout life. Apical meristems at root and shoot tips produce primary growth, increasing length. Lateral meristems, including the vascular cambium and cork cambium, add thickness through secondary growth. Plant development depends on cell division, cell expansion, and cell differentiation. Patterns of gene expression guide cells to adopt specific roles. Environmental signals also influence growth, allowing plants to adjust to changing conditions. Despite growing in place, plants exhibit complex developmental programs that produce diverse forms. Study questions. What are the three major organs of vascular plants? Answer: roots, stems, and leaves. How do apical meristems contribute to plant growth? Answer: they produce primary growth, increasing the length of roots and shoots. What is the role of vascular tissue? Answer: xylem transports water and minerals. Phloem transports sugars and other organic products. Chapter 36. Resource Acquisition and Transport in Vascular Plants. Plants must acquire water, minerals, and carbon dioxide while distributing sugars throughout the body. This chapter explains the transport mechanisms that enable these functions. Water and minerals enter roots through the soil. Root hairs increase surface area, enhancing absorption. Water moves through cells and spaces to the xylem, where it travels upward through the plant. The cohesion tension mechanism explains this movement. Water evaporates from leaves, creating tension that pulls water upward, while cohesion keeps water molecules connected. Stomata regulate gas exchange. Guard cells open and close stomata in response to environmental conditions, balancing CO2 uptake for photosynthesis with water conservation. Sugars produced during photosynthesis move through phloem in a process called translocation. The pressure flow hypothesis states that sugars are actively loaded into phloem at sources, such as leaves, creating high pressure that pushes sap towards sinks, such as roots or fruits. Plants also respond to nutrient availability. Mutualistic relationships with fungi and bacteria improve nutrient acquisition. Mycorrhizal fungi help plants absorb phosphorus, while nitrogen-fixing bacteria convert atmospheric nitrogen into usable forms. Efficient transport systems allow plants to survive in diverse habitats and support their growth and reproduction. Study questions. What drives the upward movement of water in xylem? Answer: transpiration creates tension that pulls water upward, aided by cohesion between water molecules. What is the function of stomata? Answer: they regulate gas exchange, allowing CO2 in while controlling water loss. How does translocation move sugars through phloem? Answer: sugars loaded at sources create pressure that drives sap towards sinks where sugars are needed or stored. Chapter 37. Soil and plant nutrition. Plants require nutrients from both air and soil to grow. This chapter explains how soil properties influence plant nutrition, how nutrients cycle, and how plants acquire essential minerals. Plants obtain carbon and oxygen from carbon dioxide in the air, but they rely on soil for water and mineral nutrients. Soil forms through weathering of rock and accumulation of organic matter. Its composition, including texture, mineral content, and biological activity, affects plant growth. Macronutrients such as nitrogen, phosphorus, and potassium are required in large amounts, while micronutrients such as iron and zinc are needed in smaller amounts. Nitrogen is particularly important because plants cannot use atmospheric nitrogen directly. Instead, they depend on nitrogen-fixing bacteria that convert nitrogen gas into ammonia or related compounds. Symbiotic relationships play a key role in nutrition. Mycorrhizal fungi enhance water and phosphorus absorption by increasing root surface area. Nitrogen-fixing bacteria found in root nodules of legumes and other species provide plants with usable nitrogen forms while receiving carbohydrates in return. Soil quality affects agriculture. Practices such as crop rotation, fertilization, and erosion control help maintain nutrient availability. Excessive fertilizer use can harm ecosystems, highlighting the importance of sustainable soil management. Plants are adapted to a wide range of soil conditions, and their nutritional strategies reflect evolutionary responses to resource availability. Study questions. Why is nitrogen often a limiting nutrient for plants? Answer: because plants cannot use atmospheric nitrogen and must rely on nitrogen-fixing bacteria or soil compounds. How do mycorrhizal fungi benefit plants? Answer: they increase root absorption of water and minerals, especially phosphorus. What is one way agriculture maintains soil fertility? Answer: crop rotation replenishes nutrients by alternating plant types, improving soil structure and nutrient balance. Chapter 38. Angiosperm Reproduction and Biotechnology. Angiosperms, or flowering plants, reproduce through complex interactions involving flowers, pollinators, seeds, and fruits. This chapter explains these processes and explores plant biotechnology. A flower contains reproductive organs. Stamens produce pollen, which houses male gametophytes, while carpels contain ovules that develop into female gametophytes. Pollination occurs when pollen reaches the stigma. Fertilization follows as pollen tubes deliver sperm to the ovule, forming a zygote and endosperm through a process called double fertilization. After fertilization, the ovule becomes a seed containing the embryo and stored nutrients. The surrounding ovary develops into a fruit, which aids in dispersal by wind, water, or animals. Dispersal allows offspring to colonize new areas. Seed dormancy ensures that germination occurs under suitable conditions. When conditions are favorable, seeds germinate and the embryo resumes growth. Angiosperms reproduce both sexually and asexually. Some plants produce clones through structures such as stolons or tubers. Humans use vegetative propagation and grafting in agriculture and horticulture. Plant biotechnology includes genetic engineering, tissue culture, and other methods that modify or clone plants. Genetically modified crops can provide traits such as pest resistance or improved nutrition, but they raise concerns about biodiversity, environmental impact, and food safety. Angiosperm reproduction demonstrates the adaptability of plants and the ways humans use plant biology to support agriculture. Study questions. What occurs during double fertilization? Answer. One sperm fertilizes the egg to form a zygote, while another fertilizes central cells to form endosperm. How do fruits aid in plant reproduction? Answer. They protect seeds and promote dispersal by attracting animals or enabling transport. What is one application of plant biotechnology? Answer. Engineering crops with traits such as pest resistance or enhanced nutrition. Chapter 39. Plant responses to internal and external signals. Plants constantly respond to environmental changes and internal cues. This chapter explains how hormones and signaling pathways regulate plant growth, development, and behavior. Plant hormones are chemical signals that influence processes such as growth, cell division, and flowering. Auxin promotes cell elongation and guides phototropism, bending shoots toward light. Cytokinins stimulate cell division. Gibberellins promote stem elongation and seed germination. Abscisic acid helps plants withstand drought by closing stomata. Ethylene regulates fruit ripening and responses to mechanical stress. Plants sense and respond to multiple external signals. Light affects seed germination, growth direction, and flowering time. Plants detect day length to determine when to reproduce. Gravity influences root and shoot orientation. Mechanical stimuli, such as touch, can trigger rapid movements or structural changes. Plants also defend against pathogens and herbivores. They recognize harmful microbes and activate immune responses. Chemical signals attract predators of herbivores or strengthen cell walls. Systemic responses can prepare distant tissues for possible attack. Environmental stress, including drought, salinity, heat, and cold, triggers adaptations. Some plants accumulate protective molecules or adjust membrane composition to maintain function. These signaling systems demonstrate that plants are highly responsive organisms capable of adapting to complex environments. Study questions. What hormone promotes phototropism? Answer. Auxin, which stimulates cell elongation on the shaded side of a shoot. How do plants detect seasonal changes? Answer. Through photoperiodism, sensing day length to regulate processes such as flowering. Why is ethylene important for fruit? Answer. It triggers ripening and coordinates changes such as softening and color development. Unit 7. Animal Form and Function. Chapter 40. Basic Principles of Animal Form and Function. Animal structure and function are closely linked, and this chapter explains how body plans, physiology, and environmental interactions shape the diversity of animal life. Animal form is influenced by physical laws. For example, exchange of materials such as gases, nutrients, and wastes depends on surface area relative to volume. As animals increase in size, their volume grows faster than their surface area, requiring internal systems such as circulatory or respiratory networks to distribute materials efficiently. Tissues form the foundation of animal organization. Epithelial tissue covers surfaces and lines organs. Connective tissue supports and binds structures. Muscle tissue enables movement, and nervous tissue transmits signals. Organs formed from these tissues work together in organ systems. Homeostasis maintains stable internal conditions despite external fluctuations. Animals use negative feedback loops to keep variables such as temperature, blood sugar, or water balance within ranges suitable for life. Regulators maintain constant internal conditions, while conformers allow internal values to vary with their environment. Energy use varies among animals. Metabolic rate depends on size, activity level, and environment. Endotherms generate heat through metabolism, while ectotherms rely on external sources. Both strategies involve trade-offs between energy needs and environmental constraints. Animal form and function are products of adaptation, allowing species to thrive in specific ecological niches. Study questions. Why do large animals need internal transport systems? Answer. Because their volume grows faster than their surface area, making simple diffusion insufficient for distributing materials. What are the four main types of animal tissue? Answer. Epithelial, connective, muscle, and nervous tissue. What is homeostasis? Answer. The regulation of internal conditions to maintain a stable environment within the body. Chapter 41. Animal Nutrition. Nutrition provides animals with the energy and building materials needed for survival. This chapter explains feeding, strategies, digestion, and nutrient absorption. Animals require food for energy, organic molecules, and essential nutrients such as amino acids, fatty acids, vitamins, and minerals. Different species obtain food through diverse feeding strategies, including filter feeding, substrate feeding, fluid feeding, and bulk feeding. Digestion can occur inside cells or within compartments. Most animals rely on extracellular digestion in specialized organs. The digestive process includes ingestion, mechanical breakdown, chemical digestion, absorption, and elimination. In mammals, digestion begins in the mouth, continues in the stomach, and is completed in the small intestine. Enzymes break down carbohydrates, proteins, and fats into absorbable components. Nutrient absorption occurs primarily in the small intestine, where villi and microvilli greatly increase surface area. Water and minerals are absorbed in the large intestine. The regulation of digestion involves hormones and nerves that coordinate enzyme release, muscle contractions, and appetite signals. Nutritional imbalances can lead to deficiencies or disorders that affect health. Study questions. What are the three main needs fulfilled by animal nutrition? Answer. Energy supply, organic molecules for biosynthesis, and essential nutrients that cannot be synthesized by the body. Where does most nutrient absorption occur? Answer: in the small intestine, through villi and microvilli. What controls digestive processes? Answer: hormonal and neural signals that regulate enzyme secretion, muscle movement, and appetite. Chapter 42. Circulation and gas exchange. Animals exchange gases and transport materials through specialized systems. This chapter describes circulatory systems, respiratory organs, and how gases move across surfaces. Simple animals rely on diffusion across body surfaces. Larger or more complex organisms need circulatory systems to move materials efficiently. Open circulatory systems pump hemolymph into body cavities, while closed systems use vessels to deliver blood directly to tissues. Vertebrates have closed circulatory systems with hearts that pump blood through vessels. Fish have two-chambered hearts, while amphibians and reptiles have 3 chambers, and mammals and birds have 4 chambers. These differences reflect adaptations to metabolic demand and oxygen use. Gas exchange occurs across moist respiratory surfaces. Aquatic animals use gills, which extract oxygen from water. Terrestrial animals use tracheal systems or lungs. In mammals, air travels through branching passages to alveoli, where gas exchange occurs by diffusion. Transport pigments, such as hemoglobin, bind oxygen and enhance its delivery to tissues. Carbon dioxide is transported in dissolved form, bound to proteins, or converted to bicarbonate. Circulatory and respiratory systems work together to meet metabolic needs and maintain homeostasis. Study questions. What is the main difference between open and closed circulatory systems? Answer: open systems distribute hemolymph in body cavities, while closed systems circulate blood through vessels. Where does gas exchange occur in mammals? Answer: in alveoli, where oxygen and carbon dioxide diffuse between air and blood. How does hemoglobin support oxygen transport? Answer: it binds oxygen molecules, increasing the amount of oxygen blood can carry. Chapter 43. The immune system. The immune system protects animals from pathogens such as bacteria, viruses, and parasites. This chapter explains innate immunity, adaptive immunity, and how the body recognizes and fights infections. Innate immunity provides rapid, non-specific defense. In vertebrates, this includes physical barriers such as skin and mucous membranes, as well as internal defenses like phagocytic cells, natural killer cells, antimicrobial peptides, and inflammation. These responses act quickly and do not require prior exposure. Adaptive immunity is slower to respond, but highly specific. It relies on lymphocytes, B cells, and T cells that recognize particular molecules called antigens. Each lymphocyte has receptors that bind specific antigens. When activated, B cells produce antibodies that neutralize pathogens, while T cells kill infected cells or coordinate immune responses. Adaptive immunity has memory. After an infection, memory cells persist in the body, enabling faster and stronger responses upon re-exposure. This principle underlies vaccination, which stimulates immune memory without causing disease. The immune system must distinguish self from non-self. Failure of this recognition can lead to autoimmune diseases, in which the body attacks its own tissues. Immunodeficiency disorders, such as HIV infection, weaken defenses and increase vulnerability to pathogens. The immune system is a complex network of cells and signals that together protect the body from diverse threats. Study questions. What distinguishes innate immunity from adaptive immunity? Answer: innate immunity is rapid and non-specific, while adaptive immunity is slower but highly specific and has memory. What is the role of antibodies? Answer: they bind to antigens, neutralize pathogens, and mark them for destruction. Why does vaccination work? Answer: it generates memory cells that enable a faster and stronger immune response upon future exposure. Chapter 44: Osmoregulation and Excretion. Animals must regulate water balance and remove metabolic wastes to survive. This chapter explains how different organisms manage osmotic conditions and how excretory systems function. Osmoregulation controls the movement of water and solutes between body fluids and the environment. Marine animals often face dehydration because seawater is more concentrated than their internal fluids. Freshwater animals must prevent excess water from entering their bodies. Terrestrial animals must conserve water to avoid desiccation. Excretion removes nitrogenous wastes produced during the breakdown of proteins and nucleic acids. Ammonia is highly toxic and must be excreted quickly, so aquatic animals release it directly. Urea, produced by mammals and amphibians, is less toxic and requires energy to make. Uric acid, used by birds and reptiles, conserves water but is more energy-intensive to produce. Most animals use excretory structures to regulate solutes and eliminate wastes. Flatworms use protonephridia, insects use Malpighian tubules, and vertebrates rely on kidneys. The kidney filters blood, reabsorbs valuable substances, and concentrates wastes in urine. The nephron is the functional unit of the kidney. It filters fluid, reabsorbs needed molecules, and secretes additional wastes. Hormones such as antidiuretic hormone regulate urine concentration based on the body's hydration needs. Osmoregulation and excretion are vital for maintaining stable internal conditions and supporting metabolism. Study questions. Why do birds excrete uric acid instead of urea? Answer: uric acid conserves water, an advantage for terrestrial animals with limited water availability. What is the main function of the nephron? Answer: to filter blood, reabsorb useful substances, and produce urine that removes wastes. Why must aquatic animals excrete ammonia directly? Answer: because ammonia is highly toxic and requires large amounts of water to dilute. Chapter 45: Hormones and the Endocrine System. The endocrine system uses hormones to coordinate long-distance communication within the body. This chapter explains how hormones work, how they are regulated, and how endocrine organs maintain homeostasis. Hormones are chemical messengers released into the bloodstream. They bind to receptors on target cells and regulate processes such as growth, metabolism, reproduction, and stress responses. Hormones can be water-soluble or lipid-soluble. Water-soluble hormones act on surface receptors and trigger signaling pathways inside cells. Lipid-soluble hormones pass through membranes and bind receptors inside target cells, influencing gene expression. Endocrine glands such as the hypothalamus, pituitary, thyroid, and adrenal glands release hormones in response to internal cues or external stimuli. Negative feedback loops maintain hormone levels within appropriate ranges. For example, thyroid hormones regulate metabolism, and their production is controlled by feedback between the hypothalamus, pituitary, and thyroid gland. Hormones also coordinate reproduction. Sex hormones regulate gamete production, sexual development, and reproductive cycles. In response to stress, The adrenal glands release hormones that adjust metabolism and mobilize energy. Disruptions to the endocrine system can have far-reaching effects. Hormone imbalances may result from gland disorders, tumors, or environmental chemicals that mimic hormones. The endocrine system provides a flexible and efficient way to coordinate body-wide processes. Study questions. What is the main difference between water-soluble and lipid-soluble hormones? Answer. Water-soluble hormones bind to surface receptors, while lipid-soluble hormones enter cells and bind internal receptors. How are hormone levels typically regulated? Answer. Through negative feedback loops that maintain homeostasis. Why can endocrine disorders affect multiple body systems? Answer. Because hormones influence many processes, and disruptions can alter metabolism, development, or organ function throughout the body. Chapter 46. Animal Reproduction. Animals reproduce either sexually, asexually, or with a combination of strategies. This chapter explains reproductive modes, the formation of gametes, and how animals coordinate reproduction with environmental conditions. Asexual reproduction involves producing offspring without the fusion of gametes. It includes budding, fission, and parthenogenesis. Asexual reproduction is efficient, but produces genetically identical offspring, limiting variation. Sexual reproduction generates genetic diversity by combining gametes from two parents. This diversity enhances adaptability, but requires greater energy investment. Gametes form through meiosis. In males, sperm develop continuously in testes. In females, eggs form in ovaries and mature through cyclical processes. Fertilization may occur externally, where eggs and sperm meet in the environment, or internally, where sperm are deposited inside the female's reproductive tract. Many animals exhibit reproductive cycles coordinated with seasons, food availability, or environmental cues. Hormones regulate reproductive events such as gamete production, mating behaviors, and pregnancy. In mammals, the reproductive system includes organs for gamete production, fertilization, and, in many species, development of the embryo inside the mother. Some species alternate between reproductive modes depending on conditions, highlighting the evolutionary flexibility of reproductive strategies. Study questions. What is the main advantage of sexual reproduction? Answer. It produces genetic variation, enhancing a population's ability to adapt. How does external fertilization differ from internal fertilization? Answer. External fertilization occurs outside the body, while internal fertilization takes place inside the female reproductive tract. What role do hormones play in reproduction? Answer. They regulate gamete production, reproductive cycles, and behaviors associated with mating and development. Chapter 47. Animal Development. Development begins with fertilization and continues through stages that form tissues, organs, and body plans. This chapter outlines early developmental processes and the mechanisms guiding them. Fertilization occurs when a sperm fuses with an egg, restoring a diploid nucleus and activating the egg to begin development. Early cell divisions, called cleavage, produce smaller cells without increasing overall size. The resulting structure, the blastula, undergoes gastrulation, during which cells move and reorganize into three germ layers: ectoderm, mesoderm, and endoderm. These layers give rise to all tissues and organs. Organogenesis follows gastrulation. Cells differentiate based on gene expression and position, forming structures such as the nervous system, muscles, and digestive organs. Neurulation forms the neural tube, which becomes the brain and spinal cord. Development depends on signaling between cells. Induction occurs when one group of cells influences another's fate. Morphogen gradients and gene regulatory networks shape patterns of growth and organization. In mammals, development includes implantation, placental formation, and internal gestation. The placenta mediates nutrient and gas exchange between mother and embryo. Developmental plasticity allows organisms to adjust growth in response to environmental conditions. However, disruptions during early development can lead to abnormalities. Study questions. What is the purpose of gastrulation? Answer: to form the 3 germ layers that give rise to all tissues and organs. What structure becomes the brain and spinal cord? Answer: the neural tube, formed during neurulation. How do cells become different from one another during development? Answer: through differential gene expression, influenced by signaling and positional information. Chapter 48. Neurons, synapses, and signaling. The nervous system enables communication, coordination, and rapid responses. This chapter explains how neurons generate signals, how synapses transmit them, and how communication networks regulate behavior. Neurons are specialized cells with dendrites that receive signals, a cell body that integrates them, and an axon that carries electrical impulses. Supporting cells called glia provide insulation, nutrients, and structural support. Neurons communicate through electrical signals known as action potentials. These signals result from changes in membrane potential driven by ion channels. When a neuron reaches threshold, voltage-gated channels open, creating a rapid depolarization followed by repolarization. Action potentials travel along axons without weakening. Synapses transmit signals between neurons. In chemical synapses, the arrival of an action potential triggers neurotransmitter release into the synaptic cleft. Neurotransmitters bind receptors on the next neuron, generating excitatory or inhibitory responses. Synaptic strength can change with experience, forming the basis of learning and memory. Neural signaling networks coordinate complex behaviors. The integration of signals from multiple synapses determines whether a neuron fires. Reflex pathways provide rapid responses without requiring conscious processing. The nervous system's electrical and chemical communication enables rapid and precise coordination across the body. Study questions. What generates an action potential? Answer: a rapid change in membrane potential caused by voltage-gated ion channels opening and closing. How do chemical synapses transmit signals? Answer: neurotransmitters released from one neuron bind to receptors on the next, creating excitatory or inhibitory effects. What underlies learning at the cellular level? Answer: changes in synaptic strength, such as increased neurotransmitter release or receptor sensitivity. Chapter 49. Nervous systems. Nervous systems coordinate sensory input, processing, and motor output. This chapter explains how nervous systems are organized, how brains evolved, and how vertebrate and invertebrate nervous systems operate. Simple animals, such as cnidarians, have nerve nets, loose networks that coordinate basic movements. More complex animals have centralized nervous systems with clusters of neurons called ganglia, or brains. Bilaterally symmetrical animals typically have cephalization, the concentration of sensory and neural structures at the anterior end. Vertebrate nervous systems consist of the central nervous system, CNS, including the brain and spinal cord, and the peripheral nervous system, PNS, which carries signals to and from the CNS. The PNS contains sensory neurons, motor neurons, and autonomic pathways that control internal organs. The vertebrate brain has specialized regions. The hindbrain regulates basic functions such as breathing and coordination. The midbrain processes sensory information. The forebrain includes the cerebrum, responsible for learning, memory, and decision-making. The cerebral cortex enables complex cognition in mammals. The spinal cord relays information between the brain and the body and mediates reflexes. Peripheral nerves connect sensory receptors and effectors to the CNS. The autonomic nervous system has sympathetic and parasympathetic divisions that regulate involuntary functions. Nervous systems adapt to environmental and behavioral demands. Learning involves changes in neural connectivity, while injury can sometimes lead to reorganization of neural pathways. Study questions. What is the function of the central nervous system? Answer: it integrates sensory information and coordinates responses through the brain and spinal cord. What is cephalization? Answer: the concentration of nervous structures at the head end of an animal. Which brain region is responsible for higher cognitive functions? Answer: the cerebrum, particularly the cerebral cortex. Chapter 50. Sensory and motor mechanisms. Animals rely on sensory systems to detect changes in their environments and motor systems to generate responses. This chapter explains how sensory receptors work and how motor pathways coordinate movement. Sensory receptors convert external stimuli into electrical signals through transduction. Different receptors detect specific stimuli. Mechanoreceptors sense touch and pressure. Chemoreceptors detect chemicals. Photoreceptors detect light. And thermoreceptors sense temperature. Pain receptors detect damaging stimuli. Vision relies on photoreceptor cells that respond to light. Invertebrates, rods detect low light and motion, while cones detect color and fine detail. Light signals travel through neural pathways to the brain, where they are processed into visual information. Hearing and balance involve mechanoreceptors that detect vibrations and changes in body position. Olfaction and taste detect chemicals in the environment or food. Each sense provides unique information that animals use for navigation, communication, and survival. Motor systems control muscle activity. Skeletal muscles attach to bones and contract through interactions between actin and myosin. Motor neurons stimulate contraction at neuromuscular junctions. Muscle movement is coordinated by the nervous system and refined by sensory feedback. Together, sensory and motor mechanisms allow animals to perceive their surroundings and respond effectively. Study questions. What is sensory transduction? Answer: the conversion of a physical stimulus into an electrical signal by sensory receptors. How do rods and cones differ? Answer: rods detect low light and motion. Cones detect color and fine detail. What triggers skeletal muscle contraction? Answer: motor neurons release neurotransmitters that initiate interactions between actin and myosin. Chapter 51. Animal behavior. Behavior includes all the actions an animal performs in response to stimuli. This chapter explores how genes, learning, and evolution shape behavior. Behavior results from the interaction of genetic programming and environmental influences. Some behaviors are innate and performed without learning, such as fixed action patterns. Others are learned, including imprinting, spatial learning, associative learning, and problem solving. Animals communicate through signals such as sounds, scents, body language, and light. Communication occurs in contexts such as mating, foraging, alarm responses, and social interactions. Behavior evolves because it affects survival and reproduction. Foraging behavior balances energy gain with risk. Mating systems vary widely, including monogamy, polygamy, and promiscuity. Sexual selection shapes traits that improve mating success, such as displays or ornamentation. Social behavior includes cooperation, altruism, and social hierarchies. Altruistic behaviors can evolve when individuals help relatives, increasing their inclusive fitness. Eusocial species show extreme cooperation with division of labor. Behavior is shaped by both biology and experience, allowing animals to adapt to changing conditions. Study questions. What is the difference between innate and learned behavior? Answer. Innate behavior is genetically programmed, while learned behavior develops through experience. What is sexual selection? Answer. A form of natural selection in which traits that increase mating success become more common. Why can altruistic behaviors evolve? Answer. Because helping relatives increases the overall transmission of shared genes, improving inclusive fitness. Unit 8, Ecology, Chapter 52, An Introduction to Ecology and the Biosphere. Ecology is the study of how organisms interact with each other and with their environments. This chapter introduces the scales at which ecological questions are asked and describes the major factors that shape Earth's ecosystems. Ecologists study life at multiple levels: organisms, populations, communities, ecosystems, and the biosphere. Each level focuses on different interactions, from how an individual survives to how global climate patterns influence life. Climate is one of the most important physical factors determining the distribution of organisms. Uneven heating of Earth creates patterns of temperature, wind, and precipitation. Ocean currents, mountains, and proximity to large bodies of water further influence climate. Seasonal variations result from Earth's tilt and its orbit around the Sun. Biomes are major life zones shaped by climate. Terrestrial biomes, such as tropical forests, deserts, and tundra, differ in temperature, rainfall, and vegetation. Aquatic biomes include freshwater lakes, rivers, wetlands, and marine environments, such as coral reefs and the open ocean. Each biome supports characteristic communities of organisms adapted to its conditions. The biosphere integrates all ecosystems on Earth. Human activities, including deforestation, urbanization, and greenhouse gas emissions, are altering climate patterns and affecting the distribution of species. Ecology provides tools to understand how life responds to environmental variation and large-scale changes. Study questions. What determines the distribution of major biomes? Answer. Climate patterns, such as temperature and precipitation, which are shaped by global circulation and geography. Why does Earth have seasons? Answer. Because Earth is tilted on its axis, causing different regions to receive varying sunlight throughout the year. What distinguishes aquatic biomes from terrestrial biomes? Answer. Aquatic biomes are defined primarily by salinity, depth, and water movement, while terrestrial biomes are shaped by climate and vegetation. Chapter 53. Population Ecology. Population ecology examines how populations grow, interact with their environments, and change over time. This chapter explains growth models, life histories, and factors that regulate population size. A population includes individuals of the same species living in a specific area. Population dynamics depend on births, deaths, immigration, and emigration. Ecologists study population density, dispersion patterns, and demographics to understand these dynamics. Population growth can be modeled mathematically. The exponential growth model describes populations that increase rapidly under ideal conditions. However, resources are limited, so populations usually follow logistic growth, which slows as population size approaches carrying capacity. Life history traits, such as age at reproduction, number of offspring, and parental investment, affect population growth. Species with opportunistic strategies may reproduce quickly and produce many offspring, while others invest more heavily in fewer offspring. Populations are regulated by density-dependent factors such as competition, disease, and predation, which intensify as populations grow. Density-independent factors, including weather and natural disasters, affect populations regardless of size. Understanding population ecology helps explain fluctuations in natural populations and guides conservation and management efforts. Study questions. What is carrying capacity? Answer: the maximum population size that an environment can sustain over time. Why do most populations not grow exponentially? Answer: because limited resources, competition, and environmental constraints slow growth. What distinguishes density-dependent from density-independent regulation? Answer: density-dependent factors intensify with population size, while density-independent factors affect populations regardless of density. Chapter 54. Community Ecology. Community ecology explores how species interact within communities and how those interactions shape structure and diversity. This chapter covers competition, predation, symbiosis, trophic structure, and community dynamics. Species interactions fall into several categories. Competition occurs when species use similar resources, often reducing the fitness of both. The competitive exclusion principle states that two species competing for identical resources cannot coexist indefinitely. Species may partition resources or change behavior to reduce overlap. Predation benefits predators and harms prey. Prey species evolve defenses such as camouflage, warning coloration, mimicry, or chemical defenses. Herbivory, in which animals feed on plants, also drives evolutionary adaptations. Symbiosis includes relationships where species live in close contact. Mutualism benefits both partners. Commensalism benefits one without affecting the other. Parasitism harms one while benefiting the other. Trophic structure describes feeding relationships. Energy flows from primary producers to herbivores, then to higher-level predators. Disturbances, such as storms, fires, or human actions, alter communities. Some communities undergo succession, where species composition changes predictably over time after a disturbance. Community ecology provides insights into how species coexist, how food webs function, and how disturbances shape ecological patterns. Study questions. What is resource partitioning? Answer: the division of resources by competing species to reduce overlap and allow coexistence. How do prey species defend themselves against predators? Answer: through adaptations such as camouflage, warning coloration, mimicry, and chemical defenses. What is ecological succession? Answer: a gradual change in community composition following a disturbance. Chapter 55. Ecosystems and Restoration Ecology. Ecosystems include all organisms in a community and the physical environment they interact with. This chapter explains how energy flows through ecosystems, how nutrients cycle, and how restoration efforts help damaged ecosystems recover. Energy enters most ecosystems through sunlight, captured by primary producers such as plants and algae. Energy then flows through trophic levels, from producers to herbivores, then to carnivores, and finally to decomposers. At each step, some energy is lost as heat, consistent with the laws of thermodynamics. This loss limits the length of food chains and the efficiency of energy transfer. Nutrient cycles move chemical elements through ecosystems. The water cycle distributes water through evaporation, condensation, and precipitation. The carbon cycle involves photosynthesis, respiration, and human activities such as fossil fuel burning. The nitrogen cycle depends heavily on bacteria that convert nitrogen into usable forms. The phosphorus cycle moves phosphorus from rocks to organisms and back through geological processes. Ecosystems respond to disturbances such as storms, fires, or human modifications. Some ecosystems recover naturally, while others require assistance. Restoration ecology seeks to repair damaged ecosystems by re-establishing species, improving soils, and restoring natural processes. Bioremediation uses organisms to remove pollutants, while habitat reconstruction rebuilds degraded environments. Ecosystems depend on the balance of energy flow and nutrient cycling. Understanding these processes is essential for managing natural resources and restoring ecological health. Study questions. Why is energy transfer between trophic levels inefficient? Answer: because much energy is lost as heat at each step, leaving less available for higher trophic levels. What role do bacteria play in the nitrogen cycle? Answer: they convert atmospheric nitrogen into usable forms for plants and return nitrogen to the atmosphere through other processes. What is the goal of restoration ecology? Answer: to repair and restore damaged ecosystems by re-establishing natural processes and species interactions. Chapter 56, Conservation Biology and Global Change. The final chapter addresses the challenges facing biodiversity and the global environment. Conservation biology aims to protect species, preserve ecosystems, and maintain the processes that sustain life. Biodiversity includes genetic diversity, species diversity, and ecosystem diversity. Loss of biodiversity reduces resilience, disrupts ecosystem services, and limits future evolutionary potential. Major threats include habitat destruction, invasive species, overexploitation, pollution, and climate change. Habitat loss through deforestation, agriculture, and urban expansion is the leading cause of species decline. Introduced species can outcompete native species and disrupt ecosystems. Overharvesting of wildlife has reduced many populations to critical levels. Pollution, including plastics, chemicals, and nutrient runoff, alters habitats and harms organisms. Climate change, driven by rising greenhouse gas concentrations, affects temperature, weather patterns, and sea levels. These changes alter species distributions, disrupt reproduction, and increase extinction risk. Conservation strategies include establishing protected areas, restoring habitats, managing populations, and promoting sustainable resource use. The small population approach focuses on preserving genetic diversity, while the declining population approach seeks to identify and mitigate causes of decline. Landscape ecology emphasizes preserving corridors and connectivity between habitats. Global change requires international cooperation, informed policy decisions, and public engagement. Conservation biology provides the scientific foundation for protecting the planet's biodiversity for future generations. Study questions. What are the 3 levels of biodiversity? Answer: genetic diversity, species diversity, and ecosystem diversity. Why is habitat loss the greatest threat to biodiversity? Answer: because it eliminates living space, disrupts interactions, and removes essential resources for species survival. How does climate change affect biodiversity? Answer: it alters habitats, shifts species distributions, disrupts life cycles, and increases extinction risks.